So these shared characteristics, we can also call them properties. Properties shared by both cells, the prokaryotes and the eukaryotes. So if you asked a question to give the shared property or the properties of both the eukaryote and the prokaryotic cells, you can present these ones. The DNA having the genetic component in form of DNA. Remember, viruses are the ones that will possess the RNA. Then proteins also as their structure, as the ones that support their structure. Then the cell membrane being present in both, and ATP being processed, used as the form of energy for their processes. So that is it for the properties of both the prokaryote and the eukaryotic cell. So then another thing we need to cover, which is very important, we need to know the differences between the prokaryotes and the eukaryotes. So differences between the prokaryotes and eukaryotes, I think we'll do this in a tabular form. Prokaryotes, then eukaryotes. So differences between these two. Prokaryotes, like we said, prokaryotes will have their DNA in the nucleoid region, so they don't possess a nucleus, no nucleus, in other words, no cell nucleus, so DNA is not enclosed into the nucleus. DNA is just in a region we call the nucleoid region. Yet for eukaryotes, the DNA is in the nucleus. So here we have the nucleus present, the cell nucleus present. The cell nucleus is a double membrane organelle, like we will see. Then another difference, we can talk about the size. These ones are usually around 1 to 5 micrometers in size, but the eukaryotes are always around 10 to 100 micrometers in size. That is the range we can give them. Then another difference between the prokaryote and the eukaryote, the prokaryotes will always lack, okay, these ones don't possess a mitochondria. Mitochondria, this side is absent. This side, the mitochondria is present. Remember, this mitochondria, its basic role is to synthesize energy in form of ATP, that is adenosine triphosphate, which is required for metabolic processes of the cell and any other functions of the cell to take place. Then prokaryotes don't possess the chloroplast, no chloroplast. Then for the eukaryotes, the chloroplast is present. Chloroplast is present. Chloroplast will do a basic role of synthesizing the food requirements, the different food requirements, utilizing sunlight energy. Then another difference we can give is that if you look closely at a prokaryote, it will have few cell organelles in its cytoplasm. Few cell organelles in the cytoplasm. Then this one will have many cell organelles as compared to the eukaryotes, many cell organelles as compared to the eukaryotes. This one will have a variety of cell organelles within the cytoplasm. Then prokaryotes will possess what we call a plasmid. A plasmid is present in prokaryotes, some of the prokaryotes, but in eukaryotes the plasmid is not present. So this one is absent in the eukaryotes. So these are some of the basic differences between the prokaryotic cell and the eukaryotic cell. Maybe another difference we can think of is for prokaryotes, when they are doing what we call protein synthesis, this is manufacture of a protein, their protein synthesis takes place in the cytoplasm, entirely in the cytoplasm. But for the eukaryotes, protein synthesis takes place both in the nucleus, then as well, plus the cytoplasm. So for the eukaryotes, it is divided in two ways, because protein synthesis is simply synthesis of a manufacture of a protein, if I'm to say. But for the eukaryotes, we'll have two processes of protein synthesis. That is basically transcription, and then translation, These are the two basic processes. Now in transcription, for the eukaryotic cell, this process of transcription will take place inside the nucleus. But for translation will take place in the cytoplasm. So this is a basic difference between these two cells. So in other words, protein synthesis here is taking place in the cytoplasm, but here it is divided into the cytoplasm and as well in the nucleus. It takes place also there. But again, we need to note that one of the basic difference, like the real basic difference between the eukaryotic cell and the prokaryotic cell, is that the eukaryotes, possess what we call the membrane-bound organelles. Membrane-bound organelles. So these are organelles that are covered by a double membrane. Examples, we can talk of the nucleus. Then we can as well list the endoplasmic reticulum, ER, which can be either rough endoplasmic reticulum or smooth endoplasmic reticulum, the mitochondria. Then another component also called the Golgi apparatus. So all these are membrane-bound organelles. You won't find in the prokaryotic cell, yet they are prominent in the eukaryotic cells. So this is also a basic difference. They can ask about membrane-bound organelles. So these ones are typical for eukaryotes rather than for prokaryotes. So prokaryotes don't possess these membrane-bound organelles. Maybe another difference we can think of is that for prokaryotes, we can further divide them into bacteria. We can give examples of prokaryotes. Let me have this one here. Prokaryotes, examples we can give are bacteria and bacteria. So bacteria and archaea are some of the examples of prokaryotes. Then for eukaryotes, we can the differences we say that in examples we say prokaryotes can have bacteria and archaea. Then for eukaryotes, examples we can have plant cells, which can include the xylem cells, the steroids, and even other types of cells. Then also animal cells, other animal cells can be part of the eukaryotes. Then also cells like protozoa is also part of the examples of the eukaryotic cell. So these are the basic differences between the prokaryotes and eukaryotes. But maybe another thing to take note of is that prokaryotes don't possess the mitochondria for ATP production, but they rather have a structure called the mesosome. Where which are enfoldings that increase surface area for production of the energy they will utilize in form of ATP. So that is it for the differences between the prokaryotes and eukaryotes. So halophiles, that is it for the halophiles. They under salty conditions, the extremes we are talking about. So halophiles will be found in very salty conditions. An example is cyanobacteria, like we've seen. Then another bacteria we can think of are methanogens. Methanogens, like methanococcus. We can think of this specific one, methanococcus. So methanococcus, which is also used in the synthesis, in the breakdown of, in the production of methane. So it breaks down organic matter to produce methane, which can be used as biogas, which can also be important, though it is a bacteria. Then another important methanogen is a thermophile. Another important archaebacteria is a thermophile. And this thermophile lives under extremes of temperatures. So it lives in very high temperatures. The example of this bacteria can be Thermus aquaticus. So this one lives in extremes of temperatures, very high temperatures. But this one is very important in the field of molecular biology because it can be used in synthesis of more copies of DNA artificially. We can manufacture DNA artificially using this Thermus aquaticus. So these are basically the divisions of the prokaryote bacteria, which are eubacteria and archaebacteria. Eubacteria, we say that the common bacteria we see around, we have around that have medical impact, like Salmonella typhi, Nitrosomonas, Streptomyces, but we also have bacteria, the archaea, that will survive under extremes of temperature, under extremes of conditions, like extremes of salts, which are the halophiles, then methanogens also there, then the ones that survive under very high temperatures called the thermophiles. So that is it for that classification.